Good morning, Bio 1302. This is kind of the supplemental lecture for chapters 17 and 24. It's the first kind of lesson in module 3, which is module 3 and 4 are dedicated to biodiversity. And we'll kind of start with the least complex organisms and move our way up in terms of complexity. Everything kind of covered in this is basically just giving you a brief summary of that reading guide 3.1. So you might want to watch this video as preparation for this Friday's prep quiz 4, which will cover that same 3.1 reading guide material. So this reading guide in this part of the module, as you might know from reading the reading guide and the textbook chapters, focuses on viruses and prokaryotes. So starting with kind of these most basic forms of life that we see today. Now in the Pokemon world, the only virus or bacteria that really shows up is a thing called Pokirus. It's a virus that actually is a good thing for your Pokemon. It helps kind of improve your Pokemon's stats. But in the real world, viruses and bacteria are often associated with negatives, even though, especially as we'll see in the case of bacteria, that's not always the case. But viruses, on the other hand, are usually bad things. It's definitely a timely time to talk about viruses. It's part of the reason we're doing this all virtually is thanks to a virus. And so what is a virus? So viruses are these infectious particles, and they're really, in some ways, the most basic form of life. And it's often referred to as a kind of borrowed life. So it actually is kind of a gray area between a life form and just a conglomeration of molecules. And the reason we say that is because in many cases, as you'll see, viruses don't even have DNA. They're just RNA packaged in this protein coat that can infect other organisms, and it actually relies on other organisms in order to keep propagating. Now, I think this is one of the questions in your reading guide. There are some things that that are commonly found in all viruses. So in your textbook, and in this lecture, we'll go over some differences between viruses, but there are some things that all viruses share. So first of all, they're extremely tiny, these minuscule acellular, meaning they don't even consist of cells necessarily. But what they are these packages that contain either DNA or RNA. They can infect a multitude of organisms, from as simple to bacteria all the way up to ourselves and everything in between, essentially. And they cause most of the diseases that plague our human world, as we can definitely relate to now with COVID. But even before that, when you would hear about swine flu and other viruses that cause these pandemics, a term we'll get to towards the end of this little lecture. They cannot carry out any metabolic pathway. They rely on other organisms in order to do all of their metabolism and reproduction, or quote unquote reproduction, their propagation. They don't respond to the environment. They just kind of exist. And if it weren't for hosts, they wouldn't be able to reproduce or propagate in any way. They need hosts to survive. Other than that, like I said, they rely on other cells for metabolism. They don't have all these characteristics of cells that you might be familiar with from 1301 if you took that, but basically there's no cytoplasmic membrane or cytosol within a cell, nothing like that, no organelles. They basically are just packaged little nucleic acids. Like I said, they're not cells, but they kind of have this analogous structure. When we think of a cell, we think of this sphere or ball that has all the cellular components inside. And they have a mimic of that, which is a protein coat. And within that protein coat, they have what we refer to as their viral genome. And it's this basically very small amount of nucleic acids that can either exist as DNA or just RNA. So oftentimes we associate life as this thing having DNA, but a lot of viruses don't even have that. They just have RNA. And so because based on whether it has DNA or RNA, we refer to these as DNA or RNA viruses, COVID being one of those RNA viruses. Some components that are commonly found in all viruses also include the capsid and the envelope. So the capsid is found in all viruses, and it's this shell that you often associate with the virus. I'll show you a figure here in a second. They can have various structures, but there might be some that are more familiar to you than other. But it's basically just that protein shell that has the viral genome within it. And then the envelope is something that some viruses have. So it's just this membrane surrounding the capsid itself, and it helps them infect hosts. It's actually derived from the host's cell membrane. And so you can kind of think of these viruses that have envelopes as a sort of leather face wearing, the flesh of its victims. So it'll have this structure that is basically just one of its previous host's cell membranes. And it uses that kind of as a key to enter other cells. It's kind of creepy, but neat. Viral structures can vary depending on the virus. And so in this figure 17.2 in your textbook, there are different ways viruses can look. And some of these might be familiar to you, like the adenovirus and influenza viruses. COVID, you always see the picture of it as this sort of structure with all these spikes protruding out of it. These protein spikes are characteristic of most viruses, but they also exist in other structures like these rod shapes. It's actually a helix, so it kind of spirals its way up. And then one of the most common ones that you might think of when you see viruses on TV are these almost spider-like structures, which we'll kind of go into the structure of this in person in class, but basically, it's got these tail fibers that it uses to attach itself to the cell of a host. And then this sheath is essentially just think of a needle. It'll enter the host cell and then inject the DNA, which is within the head of this, what we call a bacteriophage. And so along those lines, I was kind of getting at this idea of, the general features of how viruses themselves replicate. So first of all, they attach, and this kind of depends on whether the host will accept the virus or not. After that, it enters. These are kind of basically logical steps. It attaches to a cell, it enters, and lots of times it can be the whole virus entering, or in the case of bacteriophages, it injecting its DNA material within the cell. Synthesis in this step, the virus, the nucleic acid, so the DNA or the RNA within the virus is taking advantage of the host cell's enzymes to replicate itself. So it's the host cell is always replicating its own DNA, and so a virus essentially just hijacks that machinery. And then there's often an assembly step where the new viruses, kind of think of it as the quote unquote offspring, are assembling themselves within the cell. And then in some way, they release them and kind of replicate and depart the host cell. And these last two steps regarding how they assemble and release can be broken up into two main cycles, which are the lytic cycle and the lysogenic cycle. So I kind of think of these as a sort of like alien process versus a more insidious process, if that helps. But in the lytic cycle, what's basically happening is it's almost like a shotgun messy approach where the virus will insert its DNA or RNA into, in this case, a bacterial host. It'll undergo a replication cycle where within it, it can basically just make more of the virus. And these are essentially, think of it as almost like fully mature viruses that then just eventually burst because there's so many phages or viruses within the cell that the cell just pops and then the virus spreads from there. So it's kind of this messy process where it just makes a bunch of itself and then kaboom. In the lysogenic cycle, I say it's a little more insidious because it essentially hides itself in the host. So upon entering the cell, the viral DNA will actually kind of attach itself and integrate itself into, in this example, the bacterial chromosome. And this forms what's called a prophage. So this is just a kind of hybrid mixture of viral DNA with the bacteria's own DNA. So since it hides in there, bacteria will reproduce normally, but it will now have that viral DNA within it and basically just repeats this cycle. So the cell, the important point here within the lysogenic cycle is that the host cell survives. And so I kind of call it this more insidious process because, you know, it's just hiding within its host as opposed to just kablooey with the lytic cycle. So kind of switching gears here, your reading guide talks a lot about flu viruses and how we name flu viruses, a special class within, you know, the group virus. There are different types of viruses. We know about DNA and RNA viruses, and it's worth spending some time talking about flu viruses in particular because it's something that we just are always, you know, exposed to. And you often see a common nomenclature for viruses, and it's some combination of H and N. So you might have heard of H1N1, which was, you know, created a whole pandemic, you know, a few years ago. You know, it feels like forever ago with COVID, but before COVID, H1N1 was the big virus everyone was worried about. And the way the nomenclature comes is from the type of two glycoproteins that occur on the exterior of viruses. So there's hemagglutinin, which is getting at the H of the virus's name, and neuraminidase, which gets at the N. So the hemagglutinin is needed for binding to things like your respiratory mucosal receptors, and they help viruses enter host cells. And then neuraminidase, on the other hand, assists in budding from host cells. So these two virus or glycoproteins, hemagglutinin and neuraminidase, one helps with entry into a cell and the other helps with the exit. And there are lots of different types. I think it's 17 different hemagglutinins that viruses can have and 9 or so neuraminidase. And so based on the number, that kind of gives you a sense of the virus's nomenclature. So H1N1 would refer to subtype 1 hemagglutinin and subtype 1 neuraminidase. And so, yeah, this is just reiterating all of what I just said here. This is how the standard nomenclature of flu viruses in particular are given. Now, one thing about flu viruses, they are RNA viruses, and the RNA genomes of flu viruses are notorious for their extreme variability. And you see this often with the need for getting flu vaccines every year, right? So two processes, you know, part of the reason we need multiple viruses or vaccines is for two reasons. First of all, flu vaccines are typically a cocktail of vaccines for what scientists predict will be the predominant kind of strain of virus, but also viruses evolve very rapidly, which contributes to their extreme variability. And two main processes contribute to this. I don't know if this is talked too much on your textbook, but it's good material to know. These processes are antigenic drift and antigenic shift. So antigenic drift just describes the genetic change that's always happening in these glycoproteins. So those H's and N's, the hemagglutinin and the neuraminidase. Because there's a rapid reproductive rate in these viruses, mutations accrue and the genetic sequences of these viruses changes really rapidly. And because of these genetic changes, and rapid evolution of glycoproteins, one thing it results in is a decreased ability of our own cells to recognize them. So we see this commonly with the COVID virus. You know, we're always talking about new variants. Part of that has to do with rapid evolution of these viruses. And with every variant, there's a potential for our own host cells to kind of forget. So if we've been exposed to COVID before, a new variant might evolve enough to be incompatible with our host memory cells. And we need a compatibility in order for our host memory cells to remember that virus. So rapid evolution is one way viruses kind of evade our immune systems. That gets at antigenic drift, these changes in genetic sequences. The other possibility is antigenic shift, which you might also be familiar with. So antigenic shift refers to these introductions of viruses across species. So there might be a virus that we have and that another species has, in this case a duck, that can enter a host with a strong immune system, like a pig, which we often refer to. We see swine flu. Other examples are other birds. They have strong immune systems, but one result of this is that they can kind of harbor a lot of nasty pathogens, including viruses. And so within this system, that you can get kind of fusions of viruses, and which creates this new virus that we have no memory cells for. And so this antigenic shift, kind of when you see the term antigenic shift, try to think of cross animal transition, and that's what antigenic shift is essentially getting at. And so through these two processes, antigenic drift and antigenic shift, we can get viruses spread. And depending on the level of the outbreak, we can call these either epidemics or pandemics. So epidemics kind of refer to these, they're still large events, but they're relatively localized in the sense of not occurring globally. So in 2009, there was an outbreak in Mexico and the US, and this was the H1N1 virus that, if you remember now, over a decade ago, was what worried us. So it started off as an epidemic, kind of localized throughout Mexico and the US, but it eventually spread throughout the whole world. And this worldwide spread, this global epidemic, is just pandemic. Pan meaning, across the entirety. So in this case, across the entirety of the globe. So pandemic just is a massive or global epidemic. So just some examples of this that you probably heard of in the past. Ebola, especially since 2014, kind of localized throughout Western Africa largely. It's epidemic status because it hasn't really spread throughout the whole world. Zika was one relatively recently that was localized in Brazil, but there was worry of it spreading here, and there were some cases reported throughout Texas, but it never got that pandemic status. H1N1 at the time of this book's writing was probably the most recent pandemic. But obviously now we're these books are going to be updated to incorporate COVID, which is the current pandemic that we're living in, because that has spread globally. And so, I mentioned in terms of antigenic drift and antigenic shift how viruses can kind of spread, but it can also be broken up into kind of three main steps. So first of all, mutations can accrue in viruses, and that's what we're seeing with COVID. With the Delta variant, a mutation occurred within COVID that made it a little bit more transmissible, and this is the rate of mutation don't change in viruses, but because they reproduce so rapidly and have such short generation times, we get mutations in terms of a time scale accrue relatively quickly, just because generations don't last years in viruses, they can last hours or days. And then one way that we end up with a global pandemic is that it starts off in a small isolated human population, like we saw in the case of COVID, isolated in terms of one region of the world, and it kind of was able to gain traction within that area before it even got noticed, and by then it spread so rapidly from there, which is what happened with COVID. But the other hypothesis in terms of COVID is regards how viruses can further evolve, and that's by evolving in a completely different animal host. So almost 3/4 of new human diseases come from transmission from other animals, where they evolve in certain ways. They can't survive in human hosts until they evolve something that does allow their transmission. So the first section of this module, chapter 17, focused on viruses. The next kind of step up in terms of the complexity of life and biological diversity that's often talked about are prokaryotes. And so when we talked about the origins of life, you can kind of see why this would be the next thing we talked about. In the fossil evidence, this was the first fossil evidence. Prokaryotes are likely, in terms of organisms, the most basic organism, or first evolved organism. So some characteristics about prokaryotes: they're almost all unicellular. There are a couple of examples your book talks about otherwise, but for the most part, they're unicellular. We are multicellular organisms, and prokaryotes are unicellular. They're obviously very small as well. The largest one spans 5 micrometers. So you would never be able to see it with the naked eye. And they come in a variety of different shapes and sizes, referring to the spherical or coxi shapes that they can have, or the rod shape, bacillus shape. That's one of the vocab terms you'll want to know, or the spiral shapes that are less common than these other two, the spherical and rod shaped ones. There are also some key traits that most, but not all, prokaryotes have. The first one is a cell wall. Prokaryotes all have cell walls, which serve to protect them, maintain their shape, and prevent bursting in certain environments. Almost all cells in general, whether we're talking about prokaryotes or eukaryotes, have cell walls. Eukaryotes, the one way they differ is in what that cell wall is made of. So in eukaryotes, like us, our cell walls are largely made of either cellulose or chitin, dependent on the organism. With bacteria, bacterial cell walls are often made of peptidoglycan. So if you're in the General Bio 2 lab, you'll remember a few weeks ago we did a gram staining experiment where it's this procedure you can use to identify whether a bacteria has a thick or thin peptidoglycan cell wall layer. And peptidoglycan is just a network of modified sugars and polypeptides. So peptido getting at the peptide part of the cell wall, and glycan, think glucose or sugar. So that name is just a combination of peptides and sugars. Within, so those are gram-positive bacteria. Within gram-negative bacteria, they have a very thin peptidoglycan layer, and what they instead have a larger amount of are a sort of polysaccharide and protein-based cell wall. So they don't have as much of that sugary exterior. Some bacteria on top of the cell wall also have a capsule, which is this sort of sticky layer of polysaccharides that have lots of functions. So these capsules allow bacteria to adhere to substrates or each other. Your book talks a bit about the formation of biofilms. So capsules are essential for sticking together. They also protect the cell from things like dehydration and protect them from attacks from either other bacteria or viruses. Within or attached to the capsule are little protrusions that are used for attaching and a term that comes up in your book, conjugation, which is kind of like a bacteria's workaround for sex. They don't have sexual reproduction. They instead have conjugation. And the way they stick to things is through these fimbriae, hair-like little structures that help them attach to one another and to substrate. These longer protrusions from the capsule are the pilus or pili, appendages that facilitate conjugation. And one thing that most bacteria do, or I guess not most, about half of them do, is they exhibit a behavior called taxis, which is basically the ability to move. And when we refer to taxis, it's specifically movement towards or away from something, from a stimulus. So, for example, some prokaryotes exhibit chemotaxis, movement away or towards a chemical. Some of them exhibit phototaxis, which is movement towards or away from light. And the way they move, one of the most common ways they move is through the use of flagella, these almost tail-like structures that they can kind of use to navigate through their area. Now, I want to bring this up because it's mentioned in your reading guide. One thing that is unique to bacteria that they can do is develop these resistant cells called endospores. And endospores are kind of their end-all strategy to survival. They basically form these dormant cells that can remain viable. They'll kind of essentially shut down until the right environmental conditions reappear, at which point they'll spawn new mobile bacteria again. And these endospores can often lie dormant in suboptimal conditions like permafrost or glaciers or underground for centuries and survive these extreme conditions still. So it's an interesting strategy that has helped a lot of these bacteria survive for centuries. And this is just one way they can do this is I mentioned glaciers as well as permafrost, but another way they can do this is by actually entering these other cells. They'll form these structures that kind of protect them for centuries on end, and within them lie the endospores that essentially bring life anew. Think of the example of the phoenix. The red here would be the ashes, and from it rise the bacteria even after centuries. Now, another way you can categorize prokaryotes, we mentioned in the General Bio 2 lab, you can break them down in terms of their peptidoglycan cell walls as gram-positive or gram-negative bacteria. Another way you can do this is by their nutritional modes. So prokaryotes can be categorized by how they obtain both energy as well as carbon. So the first kind of way to break it down in terms of energy is that bacteria can, or prokaryotes can be categorized as phototrophs or chemotrophs. And that just determines how they obtain their energy. So phototrophs obtain their energy from light, whereas chemotrophs will obtain their energy from organic compounds or sometimes inorganic chemicals like hydrogen sulfide, but whether or not that, if they obtain their energy source from light, they're phototrophs. If they don't, think chemotroph. And then further, we can break it down by how they obtain their carbon. So autotrophs use inorganic carbon sources like carbon dioxide or related compounds like that. Heterotrophs will instead obtain carbon from organic compounds like living things. And so based on that, you can kind of mix and match to get the 4 types of bacteria according to their nutritional mode. So a photoautotroph, photo means it's going to get its energy from light, and auto means it's going to get its carbon from things like carbon dioxide. And then the flip side of that would be a chemoheterotroph. Chemo referring to how it gets its energy from organic compounds. Heterotroph meaning it also gets its carbon from organic compounds. And so your book has this table, and you can look at it to kind of see if there are any familiar bacteria to you and how you can break it down according to nutritional. So there are also different ways that bacteria can reproduce. One of the most common ways is by binary fission. So basically what will happen is within the bacterial cell wall, replication of the chromosomes will begin, and then when it finishes, the plasma membrane of the bacterial cell wall will just kind of split into 2. So binary fission, binary referring to the 2 cells that come as a result, fission, that release of the 2. Contrast that with fusion, which would be 2 cells becoming 1. Binary fission is 1 cell becoming 2. So prokaryotes, kind of like viruses, have considerable genetic variation. And there are 3 main factors that contribute to this. There's the fact that they can reproduce very rapidly, and they can accrue mutations. And then I'll also talk a little bit about the genetic recombination that can occur. But these first 2 points, regarding these first 2 points, rapid reproduction and mutation, like I said, prokaryotes can often reproduce by binary fission. And in this scenario, most offspring are genetically identical to the parent cell. Now, I say most offspring here because although mutations are rare, they do happen. And so these mutations are slight changes in the genetic sequence that differentiate the offspring from the parent bacteria. Now, mutations don't necessarily occur at a higher rate in bacteria, but because generation times are so short, on the order of minutes, hours, or days, they can accumulate very quickly within the population. And so one result of this is because mutations can occur relatively quickly, you get high genetic diversity. With lots of mutations come lots of differences in genetic sequences, which is the definition of genetic variability. And with high genetic diversity, this a result of this is rapid evolution. One misnomer that I like to mention here is that prokaryotes, we often refer to these as primitive life forms. But one thing to kind of keep in mind is they're not necessarily primitive. They're actually probably among the most, they've kind of won the game of evolution in terms of evolution of life on Earth. Think about it. They were here near the beginning, and they are still here thanks to their rapid adaptation to new conditions, thanks to their rapid evolution. So in some ways, we can think of them as highly evolved and winners of the big game of evolution. Now, the last point I wanted to mention in terms of the 3 factors that contribute to their genetic diversity is genetic recombination. So we often think of reproduction in this binary fission factor, where there is no sexual reproduction, but instead it's that they just asexually reproduce, and that's right for the most part. But there is still genetic recombination going on. Recombination is something we often associate with sexual reproduction, but it's not necessary. Um, and there can be this sort of recombination, this transfer of DNA across organisms by three main modes, and those are transformation, transduction, and conjugation, a term I mentioned before when I was talking about the structures of bacteria. One term that you often hear in the media is this term called horizontal gene transfer. And all this refers to is the movement of genes across species. So the transfer of bacterial DNA, this can be to a different species of bacteria, or in extreme cases to even plants and animals. So let's kind of walk through each of these: transformation, transduction, and conjugation. So in transformation, what's largely happening is some prokaryotic cells can take up and incorporate foreign DNA from their surrounding environment. The genetic code of that foreign DNA fragment is acquired by the host cell, and because of this, this is now the host's own DNA. It basically incorporated environmental DNA into its own genome, and we call this transformation, where it just gets environmental DNA from DNA that's just lying around that the bacteria incorporates into its genome. The other way, which we kind of mentioned already, is that of transduction, which is the movement of genes between prokaryotic cells by phages, those viruses. So we kind of walked through this process of how viruses can insert DNA into bacterial hosts. Well, sometimes what's within the virus DNA is DNA from another species, from a previous one of its victims, and that can be inserted within the bacteria, and then you get this this new different DNA incorporated in the bacteria. And then lastly is conjugation, where genetic material is transferred between prokaryotic cells. So in bacteria, the DNA transfer is one way. So this is one of the ways it differentiates from other forms of reproduction. In conjugation, what's happening is a bacteria will often make a mating bridge. So this involves the pilus, that anatomical structure of the bacterial cell wall I mentioned before, and it just makes this bridge where the donor bacteria can transfer part of its DNA into the host, and then that host now has its partners, quote unquote partners, DNA within it. So this conjugation process, you can kind of see how it's a little bit analogous to sexual reproduction. And one way it ends is by the rupture of this mating bridge once the DNA has transferred. So I'll leave this lecture here since it's already getting a little bit long. But yeah, if you have any questions at any time,